Welcome. This video will present a detailed walkthrough of the STREAMLINE automated machine learning tool. Before we get started, it's useful to cover some basic terminology. First, the term instance is an example or single occurrence of something in data, for example, an individual, sample, subject, or patient. Next, the term feature refers to a measurable piece of data describing some aspect of an instance, in other words, a variable, independent variable, or attribute. The term outcome is an event or metric that can be observed or measured. In other words, an endpoint, dependent variable, or class or label if we're doing classification. Next, a model is a representation or simulation of reality, and in the context of machine learning, it's an expression of an algorithm that represents patterns and that can be used to make predictions using data. And lastly, an algorithm is a set of instructions, in other words, calculations or problem-solving operations, used to complete a task. In this simplified schematic, we can see the 9 phases that make up the STREAMLINE automated machine learning framework. Given one or more target datasets, STREAMLINE starts with data exploration and processing, then imputation and scaling, then feature importance estimation, feature selection, modeling, post-analysis, comparing datasets, replication, and generating a summary report. Before going into more detail, let's talk a little bit further about some dataset terminology. This is useful because datasets are often referenced with inconsistent language. So to avoid potential confusion, we're going to review definitions of different datasets as used in the context of STREAMLINE. First, we have a target dataset. This is a whole dataset minus any replication data that has not undergone any other data partitioning and is intended to be used in the training and testing evaluation of models within STREAMLINE. We might also refer to target datasets as development datasets. One subset of a target dataset is a training dataset. This is generally a larger partition of the target dataset used to train a model. A further subset of the training dataset is a validation dataset, which here is a secondary holdout partition of a given training set used for hyperparameter optimization. This is the product of nested cross-validation being conducted within STREAMLINE. Another subset of the target dataset is a testing dataset. This is generally a smaller partition of the target dataset used to evaluate the trained model. And lastly, we have a replication dataset. This is data that is withheld from STREAMLINE throughout phases 1 through 7, but used in phase 8, otherwise known as the replication phase. This holdout replication data is used to conduct and compare model evaluations on the same set of holdout data instances, as well as to verify the replicability and generalizability of model performance on data collected from other sites or sample populations. Of note, a replication dataset should have all the features present in a corresponding target dataset. Throughout the remainder of this walkthrough, we're going to be using a recent study of obstructive sleep apnea data as an example target problem. This data comes from the Sleep Apnea Global Interdisciplinary Consortium, otherwise known as SAGIC. This dataset includes 3,111 participants recruited from sleep centers across the world, including Germany, Brazil, Iceland, the United States, Australia, Taiwan, and China. This dataset includes a binary outcome indicating the occurrence or absence of obstructive sleep apnea, based on the following clinically relevant definitions. In our first analysis, participants with an apnea-hypoxia index greater than or equal to 15 events per hour were classified as having moderate to severe sleep apnea. And in our second analysis, participants with an apnea-hypoxia index greater than or equal to 5 events per hour were classified as having mild to severe obstructive sleep apnea. This dataset also included 5 distinct categories of potentially informative features, with 85 features in total. These categories included demographic, self-reported comorbidities, symptoms, and both craniofacial or intraoral measures from photographs. In total, the original dataset was transformed into 20 different datasets, representing one of 2 possible outcome cutoffs and 10 different combinations of available feature sets in order to evaluate their predictive utility. For much of this walkthrough, we'll just be focusing on one of these datasets, identified here as dataset J, which includes all feature categories and an obstructive sleep apnea cutoff of 15 events per hour, representing the classification of moderate to severe OSA. Before running STREAMLINE, We further separated each of these 20 datasets into corresponding development and replication datasets. In each case, 70% of the original data was partitioned into a development dataset, and 30% was partitioned as a replication dataset. The development datasets would be used for training and testing evaluations, while the replication dataset would be used for replication evaluation. As mentioned, this was done for all 20 OSA datasets, where the same instances were assigned to respective replication sets within each AHI cutoff group to ensure consistency in this partitioning whenever possible. Of note, while replication data is not required to run Streamline, using replication datasets allows us to compare all models on the same set of holdout instances, which gives us a fairer comparison for picking a top-performing model. Now let's talk about getting started with Streamline. Before running Streamline on your own data, you'll need to do the following. First off, ensure that one or more target datasets that you want to analyze adhere to the input data requirements of Streamline. Second, if you have multiple target datasets, make sure the key column labels are the same across each dataset, specifically the class label, and if relevant to your data, also the instance label and match label. Next, put any target datasets to be analyzed together in a folder with no other files. If you have replication datasets for any single target dataset, you'll want to prepare these similarly. Keep in mind that a given replication dataset is meant to pair with a single target dataset. With regard to feature types, note that Streamline will automatically attempt to identify feature types, in other words, discriminating between categorical and quantitative features. And it does this based on a user-defined cutoff. However, be aware that this automated approach is imperfect, and to the best of our knowledge, there is no automated approach that can universally correctly determine feature types from dataset to dataset. Instead, we strongly recommend that users manually confirm and identify feature types in their data to get the best results from Streamline. And lastly, while Streamline comes with pretty reliable default run parameters, you'll want to consider adjusting Streamline run parameters as needed. Next, we're going to walk through this more detailed schematic of Streamline automated machine learning. We can see again the 9 consecutive phases that make up Streamline. Let's start by focusing on datasets in this pipeline. In this figure, the data being fed into phase 1 is our target data or data sets. The data being passed into phase 2 are the training and testing sets resulting from k-fold cross-validation. Note that the testing datasets are not learned from in any way in phases 2 through 5, and are only used at the end of phase 5, the machine learning modeling phase, to evaluate model performance. Also of note, in phase 5, the hyperparameter optimization element will further partition training datasets into subsequent subtraining and validation sets, in order to conduct the automated hyperparameter sweep. And in phase 8, conducting replication analysis, any available replication datasets will be used for the first time. For the remainder of this video, we'll be walking through each phase, one at a time in greater depth, using the previously described obstructive sleep apnea analysis to provide examples of Streamline outputs. Starting with phase 1, which conducts data exploration and some of the data processing. The purpose of this phase is first, to provide the user with key information about the target dataset or datasets that they wish to analyze. This is done via an initial exploratory data analysis, or EDA. Next, this phase numerically encodes any text-based feature values that may exist in the data. Then, it applies basic data cleaning and feature engineering as elements of data processing. Then this phase informs the user how the data has been changed by that data processing via a secondary and, in more depth, EDA. And lastly, phase 1 partitions the data using k-fold cross-validation. Here we can see all of these elements organized in the schematic. They'll be completed separately for each target dataset included by the user in a designated target dataset folder. Again, the first part of phase 1 is to conduct an initial EDA. So for each target dataset, the following will happen. Streamline will report data counts, including number of features, number of instances, as well as the number of instances having each of the binary class values, and the number of missing values in the dataset. It will generate a bar plot illustrating class balance or imbalance, and a histogram of data missingness across features in the dataset. It will also report and save standard pandas data summaries, including function outputs of describe, data types, and number of unique. And it will calculate and report all pairwise feature correlations, also generating a corresponding feature correlation heat map. Moving on to phase 1 data processing. For each target dataset, the following steps are completed in sequence. First, STREAMLINE numerically encodes any text-based features, since this is required in general by scikit-learn-compatible machine learning packages, which STREAMLINE utilizes. Next, the following basic data cleaning and feature engineering steps will be completed in sequence. First, instances are removed that are missing an outcome label, since these can't help in supervised learning. Second, any features that a user has specified to ignore within STREAMLINE run parameters will be removed from the dataset and not considered. Third, STREAMLINE will engineer or add missingness features to the data. For this, any feature with a missing value proportion greater than a user-defined cutoff will have a new feature added to the dataset that encodes missingness, with 0 indicating the value is not missing and 1 indicating it is missing. This allows the user to examine whether missingness itself is predictive of a target outcome. Fourth, STREAMLINE will remove features that have invariant values. In other words, all values of that feature are always the same or are always missing. Step 5, STREAMLINE will remove features that have a missingness greater than a user-defined cutoff. Step 6, instances will be removed that have a missingness greater than that same user-defined cutoff. Step 7, features will be engineered or added based on one-hot encoding for any categorical features that exist in the data. This ensures that all categorical features will be treated as such throughout all aspects of the pipeline. Of note, the original categorical feature will be removed when this happens. Step 8, STREAMLINE will remove highly correlated features from the data, again based on a user-defined cutoff. Specifically, one feature of a highly correlated pair will be randomly removed. Of note, only perfectly correlated features can be safely removed based on correlation. Removing features that are less than perfectly correlated has the potential to lead to information loss. Next, in Phase 1 Processed Data EDA, a more comprehensive exploratory data analysis is conducted on the processed data. This includes everything that was done in the initial EDA, as well as adding univariate association analysis of all features, using a chi-square test for categorical features and a Mann-Whitney U-test for quantitative features. Furthermore, STREAMLINE will generate univariate plots between individual features and outcome for any features identified to have a significant association with outcome. Bar plots will be generated for categorical features, and box plots will be generated for quantitative features. This phase also generates a processing summary, clearly identifying data summary information starting from the original target dataset and identifying any changes that occurs as cleaning or engineering elements are applied in sequence. As we can see in this example focused on dataset J from our obstructive sleep apnea study, this particular dataset remained unchanged throughout processing, yielding the same original and processed data counts. This makes it clear to the user how their data may have changed through STREAMLINE automated data processing, and what elements were involved in changing the data. Before moving on, let's briefly talk about overfitting and sample bias in the context of data partitioning. In setting up a machine learning analysis pipeline, some type of data partitioning is essential to evaluate a model and help avoid overfitting. Overfitting is defined as fitting a model too closely to the training data such that it does not perform as well on other holdout instances. Another way of putting this is that a model does not generalize well to other instances. Here we can see a simple illustration of modeling a binary classification task, where the red line illustrates the decision boundary learned by a machine learning algorithm. The example on the left is more or less an ideal model fit to this data, while the example on the right illustrates overfitting of the training data. However, it's important to be aware that a single partition of the data into a training and testing set can just by chance result in sample bias. Sample bias can be defined as biased training or evaluation of a model on data that's not truly representative of the target population to which the model is intended to be applied. This brings us to the last part of Phase 1, which is cross-validation data partitioning. STREAMLINE employs experiment-wide k-fold cross-validation partitioning, where k indicates the number of partitions. This means that ultimately, STREAMLINE will train and evaluate k models for every machine learning modeling algorithm being run. To briefly review k-fold cross-validation, we start with a target dataset, which is stratified into k folds, or in this example 5 folds. We then form k training and testing datasets as separate splits of the data, where in each split, a different fold is designated as the testing set, and the remaining folds make up the corresponding training set. And again, each split will be used to train a separate model, which can be evaluated by its corresponding testing set. And later, we can evaluate model performance by taking the average testing evaluation across all K models that were trained. This type of partitioning is suited to more fairly comparing algorithm performance by better accounting for potential sample bias. Of note, Streamline uses 10 partitions by default, but users can specify other values of K. Also, Streamline includes 3 separate cross-validation options as a user run parameter. The first is stratified cross-validation, which is used by default. This type of partitioning randomly splits the target data, but also seeks to maintain class balance between the folds. Streamline also includes random partitioning as an option, as well as group partitioning, which is similar to stratified partitioning, but also keeps instances that have the same designated match group together within a single fold. This is useful for analyzing data that has been covariate matched to remove covariate effects. Also of note, Streamline saves all cross-validation datasets as CSV files to help ensure reproducibility and for users to apply externally as desired. Next, we move on to Phase 2, which conducts imputation and scaling. Importantly, this phase conducts the data preparation steps that must occur after cross-validation partitioning in order to avoid data leakage as a potential bias. Imputation takes place first to avoid scaling issues in the presence of missing values. Both steps are trained and applied separately to each individual training dataset, and testing datasets are not looked at when imputation or scaling is training in order to avoid data leakage. Once imputation and scaling have been trained, they are applied in the same way to the testing dataset without learning from the testing dataset. Of note, both imputation and scaling can be turned off as options, but imputation must be on when missing data is present in order to run most scikit-learn modeling algorithms in Phase 5. And scaling should be on for certain modeling algorithms to learn effectively, for example, artificial neural networks. And it should also be on if users want to be able to infer feature importances directly from certain algorithms' internal estimators, which for example would be the case for logistic regression. Here we can see Phase 2 is part of the schematic. For imputing missing values, mode imputation is used for categorical features, and then an iterative imputer is used by default for quantitative features. And scaling utilizes a standard scaler to transform feature values based on the mean and standard deviation of that feature in the training data. Of note, Streamline saves the learned imputation and scaling procedures for later use. Also of note, for larger datasets, multivariate iterative imputation can be slow and require a great deal of memory. As such, the user can deactivate multiple imputation and just use median imputation for quantitative features instead. Before moving on, let's talk a little bit more about scaling. As mentioned, the standard scaler is used to transform features to have a mean of 0 as well as unit variance. In general, the primary drawback to scaling prior to modeling is that any future or replication data applied to the model will need to be scaled in the same way prior to making predictions. Fortunately, Streamline does this automatically for any downstream replication data. Also of note, any algorithms that have a directly interpretable model, for example decision trees, the values specified by these models will need to be unscaled in order to understand the model in the context of the original data values. Again, fortunately, Streamline includes a useful notebook that can generate these direct model visualizations for decision trees and genetic programming models. And this code automatically unscales the values specified in these models so they retain their interpretability. Now we'll move on to Phase 3, which conducts feature importance estimation. This phase applies fast feature importance estimation algorithms, which are often used as filter-based feature selection algorithms. These include mutual information, which scores features based on univariate associations with outcome, and multisurf, a relief-based algorithm that scores features in a way that is sensitive both to univariate and epistatic or multivariate feature interactions. In Streamline, both algorithms are run by default, however the user can optionally deactivate either. And for datasets with a larger number of features, on the order of 10,000 or more, we recommend turning on the turf wrapper algorithm as a user option, which has been shown to be able to improve multisurf sensitivity to interactions in data with a higher number of features. Overall, Phase 3 is important firstly to drive the subsequent feature selection phase, but also as an opportunity to evaluate feature importance estimates prior to modeling. as well as outside the initial univariate analysis conducted in Phase 1 on the entire target dataset. It's also an opportunity to contrast feature rankings between mutual information and multisurf, which can highlight features that have little or no univariate effects but are involved in epistatic interactions. Here we can see Phase 3 within the schematic. This phase also outputs ranked bar plots of feature importance estimates. On the left, we have the bar plot for mutual information, and on the right, we have the bar plot for multisurf. Moving on, let's talk about Phase 4, which conducts feature selection. This phase uses the feature importances learned in Phase 3 to conduct feature selection, using what's called a collective feature selection approach. By default, STREAMLINE will remove any features from the training data that scored 0 or less by both feature importance algorithms, as generally, such features are deemed uninformative. Users can optionally ensure retention of all features prior to modeling by turning off feature selection. Users can also specify a maximum number of features to retain in training datasets. Doing this can help reduce overall pipeline runtime and make learning easier for modeling algorithms, assuming the feature importance algorithms were successful in capturing the most important features. To explain the collective feature selection process, if after removing uninformative features, there are still more features present than the user-specified maximum, STREAMLINE will pick the unique top-scoring features from one algorithm, then the other, until this maximum number of features is reached, at which point all other features will be removed from the training dataset. And any features identified for removal from the training set will consequently be removed from the testing dataset as well. Here we can see Phase 4 feature selection within the STREAMLINE schematic. At this point, we have K training and testing datasets that have been processed, scaled, imputed, and have undergone feature selection. Of note, while Phase 1 data processing impacts all K datasets in the same way, scaling, imputation, and feature selection can be accomplished differently within each of the K dataset pairs. If you want to learn more about the collective feature selection approach utilized by STREAMLINE, check out this paper published in 2018. Now let's take a look at the results of collective feature selection on our example obstructive sleep apnea dataset J. In our example study, collective feature selection was applied to 10 cross-validation datasets, each with 85 total features. STREAMLINE automatically generates this table, identifying the number of features that were deemed informative versus uninformative within each cross-validation partition. In this case, the number of informative features is the total number of features that will be passed onto machine learning modeling within each cross-validation partition. Similar to earlier, new fully processed and feature-selected cross-validation datasets are saved for future or external use. And downstream replication analysis will similarly select the same feature set from the replication data prior to model reevaluation. Before moving on to modeling, let's briefly talk about the no free lunch theorem. In short, this states that any two optimization algorithms are equivalent when their performance is averaged across all possible problems. Another way of thinking about this is that there's no single algorithm that can be expected to perform optimally across all problems or datasets. Or even more simply, that sadly there's no one ring to rule them all. However, this does not mean that algorithm choice doesn't matter. But it does mean that you shouldn't always use the same algorithm for every problem. And this lesson also applies to hyperparameter selection within a given algorithm, in that you don't always want to rely on the same or default hyperparameter settings when running any algorithm. This brings us to Phase 5 of STREAMLINE, which conducts machine learning modeling. First off, users will want to decide what machine learning modeling algorithms to run, as well as whether to limit the number of training instances that can be used by more computationally expensive algorithms. In summary, this phase includes hyperparameter optimization, which is conducted using training data, training final models using optimized hyperparameter settings, which is also conducted using the training data, calculating model feature importance estimates, also based on the training data, and evaluating model performance across 16 standard classification metrics, instead using the testing data for the first time. Here we can see Phase 5 focused on machine learning modeling within the STREAMLINE schematic. The 16 machine learning algorithms currently included in STREAMLINE are naive Bayes, logistic regression, elastic net, decision tree, random forest, gradient boosting, XGBoost, LGBoost, CatBoost, support vector machine, artificial neural networks, k-nearest neighbor classifier, genetic programming, and 3 rule-based machine learning implementations developed by our lab, including ELCS, XCS, and extracts. Also, users with a little coding experience can fairly easily also add other scikit-learn compatible algorithms to this repertoire. This phase automatically completes the following steps in sequence: a hyperparameter optimization sweep, training a final model on the entire training set using the best hyperparameter options identified in the hyperparameter sweep, calculating model feature importance estimates, and performing model performance evaluations on respective testing datasets. We'll talk about each of these steps in a bit more detail in the slides that follow. First, let's talk about model selection. As mentioned, STREAMLINE currently includes 16 scikit-learn compatible machine learning modeling algorithms. By default, STREAMLINE is currently set up to use 14 of these, excluding XCS and ELCS, which are both experimental implementations of existing algorithms developed by our lab. These are most likely to be of greatest interest to the rule-based machine learning research community. Users can decide to run anywhere from 1 to 16 algorithms at once within the STREAMLINE framework. And as mentioned, users can also add their own scikit-learn compatible algorithms. STREAMLINE uniquely includes 3 rule-based machine learning algorithms, also known as learning classifier systems, or LCSs. As mentioned, these include extracts, XCS, and ELCS. Previous work has shown that learning classifier systems are able to detect complex associations while also providing human-interpretable models in the form of if-then rule sets. Extracts was specifically designed to handle the challenges of scalability, noise, and the detection of epistasis and genetic heterogeneity in biomedical data mining. If you want to learn more about extracts, check out this paper published in Evolutionary Intelligence in 2015. In general, we recommend using at least the following algorithms when running STREAMLINE: naive Bayes, logistic regression, decision tree, random forest, XGBoost, support vector machine, artificial neural networks, and extracts. We have found these to be a reliable set of algorithms with an array of complementary strengths and weaknesses on different problems. Now we'll talk about Phase 5 hyperparameter optimization. Most machine learning algorithms have a variety of hyperparameter options that influence how the algorithm runs and performs on a given dataset. STREAMLINE adopts the Optuna package to conduct an automated Bayesian optimization of hyperparameters. We've set up STREAMLINE with a comprehensive range of possible important hyperparameter settings that have been hard-coded for all algorithms. An exception to this is naive Bayes, which does not have any hyperparameter options. The benefit to having these hard-coded is that users don't need to specify hyperparameter options or ranges. However, advanced users can certainly update these values within the code before running STREAMLINE. Of note, learning classifier system algorithms can be computationally expensive in contrast to some of the other machine learning algorithms included. However, they have largely reliable default run parameters. Therefore, hyperparameter optimization is turned off for these algorithms by default, but they can be turned back on if desired. As mentioned earlier, on whole, STREAMLINE adopts a nested cross-validation approach, given that within hyperparameter optimization, training datasets are further partitioned into sub-training datasets and validation sets to evaluate hyperparameter options without looking at the testing data. Within STREAMLINE, Optuna automatically further partitions a given training dataset with threefold cross-validation, yielding 3 training and 3 validation sets to evaluate hyperparameter configurations. While in a separate video we'll cover STREAMLINE run parameter options in more depth, we'll briefly talk about hyperparameter options here. The first is the evaluation metric used to conduct the optimization. STREAMLINE uses balanced accuracy by default. Next, we have the metric direction, indicating whether we want to maximize or minimize the target evaluation metric. And for balanced accuracy, we want to maximize it during optimization. The number of trials option indicates how many hyperparameter option combinations will be tested during the sweep, which is set to 200 by default, mainly to reasonably limit STREAMLINE runtime. Furthermore, the timeout option indicates the maximum allotted time for the hyperparameter sweep as a whole, after which point it will stop searching even if it hasn't reached the desired number of trials. Furthermore, STREAMLINE includes separate fixed learning classifier system hyperparameters that can be directly specified by users, including key learning classifier system run parameters of population size, number of learning iterations, and accuracy pressure, with default values shown here. Regarding hyperparameter optimization output, STREAMLINE stores the optimized hyperparameter settings for each algorithm and cross-validation partition for future reference. and Streamline can optionally generate Optuna visualizations of the hyperparameter sweep that was conducted to get a sense of how different hyperparameters impact performance on the given dataset. Once the hyperparameter sweep is completed, every model is retrained with the identified optimized hyperparameter settings. And it does this using the entire respective training set, and all models are saved as pickled files for future use. Turning now to Phase 5 model feature importance estimation. It's important to note that this is distinct from the initial Phase 3 feature importance estimation, in that these estimates are specific to a given model as a useful part of model interpretation and explanation. By default, Streamline employs permutation feature importance for estimating feature importance scores in the same uniform manner across all algorithms. Of note, some machine learning algorithms have a built-in strategy to gather model feature importance estimates. These algorithms include logistic regression, decision tree, random forest, XGBoost, LGBoost, gradient boosting, ELCS, XCS, and extracts. Users have the option to use these internal estimates instead of the permutation feature importance ones. This will direct Streamline to report any available internal feature importance estimates for a given algorithm, while still utilizing permutation feature importances for algorithms with no such internal estimator. The last part of Phase 5 is model evaluation using 16 established classification metrics. These include the 4 most basic classification metrics: false negatives, true positives, false positives, and true negatives. These 4 metrics can be used to calculate an array of other standard classification metrics, including sensitivity and specificity, precision and negative predictive value, standard accuracy, balanced accuracy, and F1 score, and the positive and negative likelihood ratios. Additionally, the area under the ROC curve is reported, as well as the area under the precision recall curve and the average precision score. Moving on to Phase 6 of Streamline, which conducts post-analysis steps. Here, mean and median cross-validation performance is reported for all classification metrics and model feature importance estimates. Also, statistical comparisons of algorithm performance are conducted, and the majority of results visualizations, including ROC and PRC plots, composite feature importance plots, and box plots of both metric evaluations and feature importance estimates. This phase ends by saving and organizing all raw and summary evaluation files. On the whole, this phase combines, summarizes, analyzes, and visualizes all modeling results. The next several slides will take a look at the different outputs generated in this phase. The most basic output is the summary of evaluation metrics across all algorithms. This automatically generated table gives median algorithm performance across tenfold cross-validation models for each of the 16 classification metrics. Within each metric column, notice that the best performing algorithm is automatically highlighted with a gray cell. Already in this example, we can see that the choice of evaluation metric can change our interpretation of which algorithm performed best. Streamline generates a similar table for mean algorithm performance. Next, we have the ROC plot, comparing average algorithm performance in contrast with a dotted no-skill line, representing performance no better than random chance. An ROC plot is most appropriate for evaluating model performance when class counts are reasonably balanced. We also have a precision recall plot, comparing average algorithm performance in contrast with a dotted no-skill line, again representing performance no better than random chance. Note that the position of the no-skill line in PRC plots can be different depending on the ratio of positive to negative class instances in the data. This example dataset happens to be largely balanced, putting the no-skill line close to 0.5. A PRC plot is more appropriate to focus on if there is more significant class imbalance in a dataset. Streamline also generates ROC and PRC plots for each individual algorithm, showing performance across all data partitions, as well as mean performance as given comparing algorithm performance in the previous slide. Streamline also generates box plots, comparing algorithm performance on each of the cross-validation datasets. Here we have the box plot for the balanced accuracy metric. And here's one for area under the ROC curve, and one for area under the precision recall curve. These box plots are generated for all classification metrics and algorithms by default. Next, we have model feature importance box plots generated for each algorithm. The one shown here is for the extreme gradient boosting algorithm. By default, these plots illustrate the top 40 features ranked by mean feature importance across each of the cross-validation datasets. The last post-analysis visualization we'll talk about is the composite feature importance plot. This plot combines the mean feature importance scores across all algorithms after normalization and weighting these scores by respective algorithm performance, in this case the balanced accuracy metric. This plot is useful for looking for consensus among the top performing algorithms as to what the most important features were in modeling, as well as to compare and contrast what different algorithms viewed as being the most important features. The last post-analysis element we'll talk about are the statistical comparisons. For simplicity, STREAMLINE uses non-parametric statistical significance tests. The Kruskal-Wallis test is used to assess whether there's a significant difference for each performance metric among all algorithms. Here we can see some example results indicating that there were some significant differences among algorithms for all 16 metrics in this example analysis. Subsequently, both the Wilcoxon rank test and Mann-Whitney U tests are applied for any metric that yielded a significant Kruskal-Wallis test. For both of these metrics, respective pairwise statistical tests between algorithms are then conducted to identify which algorithms perform significantly better than others. Moving on to phase 7 of STREAMLINE, this phase conducts dataset comparisons. This phase should be run when STREAMLINE has been applied to more than one target dataset at once. Similar to the last phase, it applies non-parametric statistical significance testing between datasets to identify if performance differences were observed. This includes comparisons of dataset performance using only the best performing algorithms for each target dataset, as well as comparisons of dataset performance on an algorithm by algorithm basis. This phase automatically generates plots to compare performance across datasets, as well as examine algorithm performance consistency. Here we have a box plot comparing mean balanced accuracy across all algorithms and datasets. The 20 datasets on the x-axis of this plot are our 20 separate example obstructive sleep apnea datasets, using the 2 different AHI cutoffs to define class outcome and the 10 different combinations of feature categories. The colored lines for each algorithm show that algorithm's mean performance within each dataset. Here's a similar plot for the F1 score evaluation metric, and another for the ROC area under the curve metric, as well as the PRC area under the curve metric. One of the many observations in the results of this analysis is that OSA datasets using a cutoff of AHI greater than or equal to 5 achieve a significantly higher area under the precision recall curve than the corresponding datasets using the AHI greater than or equal to 15, meaning that this data was more useful to identify patients with any degree of obstructive sleep apnea than those with moderate to severe obstructive sleep apnea. To further illustrate how these dataset comparison plots can be useful, here are similar plots from 2 separate simulation studies published in the first STREAMLINE paper. In this first plot, we have an analysis of 6 simulated genomic datasets, each with a different underlying pattern of association. The first 2 datasets have simple univariate or additive relationships, and we can see that pretty much all machine learning algorithms perform about the same on each. The third dataset introduces a heterogeneous association, leading to a greater degree of algorithm performance spread. And this algorithm performance difference becomes most dramatic in dataset 4, which introduces a pure 2-way epistatic interaction between features. And the 5th and 6th datasets representing increasingly complex underlying relationships in the data. In this second plot, we have 6 datasets representing a well-known computer science benchmark problem known as the multiplexer problem that increases in complexity from left to right datasets. Each of these datasets requires the algorithm to find an increasingly complex combination of feature interactions and heterogeneous associations, again allowing us to easily see the benefit of considering a variety of algorithms when analyzing any new dataset. Moving on, this phase also generates algorithm-specific box plots comparing ROC and PRC area under the curve across all datasets. These plots show performance differences using the artificial neural network algorithm for modeling. And lastly, this table illustrates just a part of the statistical significance testing between datasets conducted in this phase. These analyses are automatically output as CSV files for the user to examine. Next, we have phase 8 of STREAMLINE, which conducts replication. This phase should be run when you have some externally held out or newly acquired replication data. Here, one or more replication datasets can be used to reevaluate all models trained on a given target dataset. Of note, this phase would need to be run multiple times to reevaluate the models of each target dataset if multiple datasets were run in Streamline at once, where each target dataset has its own replication dataset or datasets. Of note, this phase would be the first time that trained models were evaluated and compared on the same set of instances. This makes it preferable for picking a best performing model or further evaluating model generalizability and its ability to replicate performance on data collected at different times, sites, or populations. The replication phase starts by conducting initial EDA on each replication dataset, much like in phase 1. This phase also processes each replication dataset in the same way as its original corresponding target and training datasets, including cleaning, feature engineering, missing value imputation, feature scaling, and feature selection. And this process will yield the same number of features, but not necessarily the same number of instances as its corresponding training dataset. And again, the EDA is repeated on processed replication data to confirm consistent processing of that replication data and to generate a respective feature correlation heat map. Note that univariate analysis is not conducted on the replication data. Next, all models previously trained for the target dataset are reevaluated using each processed replication dataset and across all classification metrics as in phase 5. These results are organized and saved separately in the user-specified experiment output folder. And all model evaluation plots are automatically generated, including ROC and PRC plots and metric box plots. Note also that no feature importance plots are generated currently in this phase, and non-parametric statistical tests are applied to examine differences in algorithm performance. Here we can see the ROC plot comparing average algorithm performance, now evaluated on the replication data. This is in contrast with the original plot evaluated on the corresponding testing dataset. And lastly, phase 9 of Streamline generates a summary report. Such a report can be generated for both testing, capturing phases 1 through 7, or for each replication analysis, capturing phase 8. These PDF reports provide a convenient, easily shareable documentation of the analysis, including Streamline run parameter settings and major results for all analyzed datasets. That concludes this detailed walkthrough of the phases completed by the Streamline automated machine learning pipeline. These phases have been arranged not only to clearly explain how this pipeline works, but also to encapsulate the most efficient arrangement of the pipeline when running it using high-performance computing parallelization. Thanks for listening to this detailed walkthrough of Streamline AutoML. Please feel free to reach out if you're interested in collaborating or if you have any questions.